Hello everyone, I am Dr. Priyanka Saddevi here, NEET PG educator on an academy. Today I am here to teach you a small topic from microbiology but yet very important topic, that is acid fast stain, also known as Zeal-Nelson stain. Many MCQs are framed from this small topic. So what I will teach you in this, it is a small lecture. I will teach you the method of acid fast stain, principle of acid fast stain, interpretations, principle, interpretations, method, and how it is different from gram stains, and examples of organisms which show acid fast stain apart from mycobacilli. So I will give you mnemonic also. So without wasting time, I am just starting it. So acid fast stain, also known as Zeal-Nelson stain, it is discovered by a scientist or person known as Ehrlich, and it is modified by Zeal and Nelson. See the spelling of Zeal and Nielsen. Zeal and Nielsen. It is modified by Zeal and Nielsen. It is used for staining a bacteria known as mycobacteria. Mycobacteria tuberculosis also, and mycobacteria Lipri also. Both the mycobacteria can be stained from it. Now, why only this mycobacteria gets stained of the Zeal-Nelson stain, acid fast stain, not other bacteria are staining? What is the difference? The difference lies in the structure of the cell wall. You can see three types of bacteria are there in this diagram. You can see gram-positive organisms have thick cell wall having peptidoglycan in their cell wall. So gram-positive have a thick layer of peptidoglycan in their cell wall. That's why they are gram positive and they take the primary stain in gram staining, that is the violet stain, gentian violet stain. These are the gram negative organisms. In their cell wall you can see a thin, very thin layer. They have a very thin layer of cell wall, of peptidoglycan in their cell wall, and apart from it they have an outer membrane also. They have an outer membrane which is absent in the gram positive organism, and that is the reason these are gram negative. They do not take the primary stain in gram staining. They took the secondary stain, that is carbon fuchsin, that is they look red. They look blue or violet in gram staining. In gram staining they look red. Now what is difference in acid fast organism, or what is different in mycobacterium bacillus as compared to other gram positive and gram negative bacteria? Look, in the cell wall they have a special fatty acid, special lipid. The name of the fatty acid or lipid is mycolic acid. This mycolic acid is present only in cell wall of mycobacteria. That is absent in other bacteria. Other bacteria cell wall made up of peptidoglycan. Here also a thin layer of peptidoglycan is present. You can see I am marking with blue. But apart from it, they have mycolic acid also. So what is the method of doing the acid fast stain? First take the primary stain. Primary stain here is carbol fuchsin, which is a red color stain. So red color stain is given. Carbol fuchsin will enter inside the bacteria and provide red color to it, and it will enter inside only those bacteria which have mycolic acid in their cell wall, that is mycobacteria. So carbol fuchsin is the primary stain. After that we heat. It is not written here, right? Heat. We will heat the slide. Heat is acting a moderant here, that it is fixing the dye. Meaning of moderant means it will fix the dye inside the bacteria. So after that heat, the third step we use a decolorizer. Decolorizer standard use here is 20% sulfuric acid. Although we make many modifications, I will come on modifications also, but standard decolorizer used in acid fast stain is sulfuric acid. It is 20%. Please see the concentration also. It is standardly it is 20% sulfuric acid. And the last step we use a counter stain, secondary stain or counter stain. The counter stain is methylene blue and it is blue or violet in color. It is blue. So primary stain is red in color, and the secondary stain is blue or violet in color. So some bacteria retain the primary stain, that is carbol fuchsin. They resist decolorization. During decolorization step, they are not decolorized. They retain primary stain only. They resist decolorization. These are known as acid fast organism. And there are some bacteria who do not resist decolorization. They got decolorized by 20% sulfuric acid. So they took the secondary stain, that is methylene blue, and they look blue, not red. These are known as non-acid fast. So this is the procedure. See, everyone see here. I will tell you, initially all organisms are transparent. Can you see here? This, I am taking two bacteria. This is also transparent. This is also transparent. I am putting mycolic acid, carbol fuchsin in both of them. That is primary stain. So primary stain is taken by both of them. This also bacteria taking primary stain, becoming red. Carbol fuchsin is red in color. And this bacteria is also taking primary stain and looking red in color. So both of them become red. Then I will heat both of them. Heat will act as a moderant. After that, I will put the decolorizer. The decolorizer is 20% H2SO4, sulfuric acid. So some bacteria are resisting decolorization. They resist decolorization. They don't decolorize. They retain. They resist decolorization. Number one. That's why they retain primary stain. Number two. Opposite is done in some other bacteria. They do not resist decolorization. They got decolorized. They do not resist. So they lose, instead of retaining, they lose primary stain and they become transparent again. They become transparent. The last step, we are putting counter stain or secondary stain, which is of blue color, methylene blue. So the first bacteria which was resisting decolorization, they contain primary stain only. They contain only primary stain, so there is no space inside them. They do not take secondary stain. They will remain red only at the end of the procedure. But the another bacteria which in which the primary stain is lost and decolorization is done, now they will take the secondary stain and they will look blue. So the red bacteria, in the end the bacteria is red. It is known as acid fast bacteria, and the bacteria which is blue in the end, it is known as non-acid fast. Non-acid fast. What do you mean by fastness? Fast, acid fast and non-acid fast. What do you mean by fastness? Now please listen. Fastness means resistance. Fastness is equal to resistance to decolorization. Resistance to decolorization. As I am saying, these bacteria are acid fast, so they resist decolorization. That's why retain primary stain. And these are not fast. These are not acid fast, so they do not resist decolorization. They do not resist decolorization and they got decolorized, and that's why primary stain is not retained and they took the secondary stain. I hope I am clear to you. So the same is seen here also. See, the 4 steps are in front of you. The first step is adding primary stain, that is carbol fuchsin, which is red in color. Second step is heating act as a moderant. The third step is putting decolorizer. Decolorizer here is 20% H2SO4. And the last step is putting counter stain. So counter stain is blue in color and primary stain, that is carbol fuchsin, is red in color. So the 4 steps are in front of you. See, take a slide. On the slide, your smear is present. On the smear, you are putting the primary stain. The primary stain is red in color. You are putting via dropper carbol fuchsin. So all bacteria, can you see? Some bacteria are circular, some are elongated. Some are cocci, some are bacilli. I am marking. So all of them took the red color and all the bacteria on the slide become red. They took the carbol fuchsin. Carbol fuchsin is the primary stain. Now after that you are heating. Can you see? Over the flame or over the steamer, you are heating it. So heat here act as a mordant. Heat, it is fixing the dye. The third step, you are putting decolorizer, which is 20% sulfuric acid drop by drop over the same slide. So some bacteria, can you see, these one become transparent. If you zoom it out, see, these are the transparent bacteria. Some of them lose the primary stain, they become transparent, and some of them retain the primary stain, they resist decolorization. So in short, some are resisting decolorization, becoming red only, and some are not resisting decolorization. They are losing the red color, they become transparent again. Now the last step, you are putting counter stain, which is blue in color. A counter stain here is methylene blue. So methylene blue is the counter stain. Primary stain was carbol fuchsin. So see the dropper, the blue color, drop by drop you are putting over the same slide. So the bacteria which become transparent, they will take the secondary stain now. They will take the secondary stain and they will appear blue. Can you see? जो जो वहाँ पे transparent हो गए थे वो अब यहाँ पे blue हो गए. और जो वहां पे ट्रांसपेरेंट नहीं हुए थे, they are red, still red, because inside them there is no space for secondary stain. They will retain primary only. So two colors of the bacteria is obtained at the end. So the red one are acid fast and the blue one are non-acid fast. So the red one are the bacteria which resist decolorization, means they are fast. Fastness is there, resistance is there. So they retain primary stain. The non-acid fast are the bacteria which are not resisting the decolorization. So these are not acid fast. Not acid fast. So they will tip the color of secondary stain. I hope I am clear in the principle and why. Now the question arises, why some bacteria are retaining primary and do not undergoing decolorization and some are undergoing decolorization. The answer to this question is mycolic acid in the cell wall. The bacteria which have mycolic acid in their cell wall, they, once the carbol fuchsin go inside them, carbol fuchsin cannot come out of them. It will become permanently trapped inside them because of mycolic acid in the cell wall. It is a type of lipid or fatty acid in the cell wall. The same thing, the same story. First step, carbol fuchsin. So both the bacteria become red. Beautiful diagram. Both, see the two bacteria. Both of them become red. Second step, heat is not shown here. Second is heat. Third is decolorization. Decolorization by 20% H2SO4. Some bacteria retain the primary stain and they resist decolorization. Some of them lose primary stain. They are doing decolorization, so they become transparent again. So last is methylene blue. Last step is the addition of counter stain methylene blue. So the bacteria which was retaining primary, there is no space for methylene blue, they will remain red only. And the bacteria which become transparent, they will take this methylene blue and they become blue. So in the end, two colors, red, blue. Red are acid fast, blue are non-acid fast. So have you got the principle? So what is the primary stain used? Tell me the answer. It is concentrated carbol fuchsin. What is the decolorizing agent? Mostly it is 20% sulfuric acid, but sometimes it is 5% sulfuric acid also. In which scenario I will tell you. And what is the counter stain? Counter stain here is methylene blue. So method, I have already discussed the method. Take a slide, fix your smear, put carbol fuchsin on it, gentle heat over the flame, and carbol fuchsin remain there for 5 to 10 minutes. And heating, you just heat it. After 5 to 10 minutes, wash the slide. So after 5 to 10 minutes, then heat it, then 5 to 10 minutes, wash. You have to wash the slide. After washing, put 20% sulfuric acid over it, and it is repeated till the pink color stops coming out. In case of Lipra, use 5% sulfuric acid instead of 20. You can also use acid alcohol instead of sulfuric acid, and then keep it there for few seconds. After that, put the methylene blue, which is a counter stain, for next 2 minutes, and then wash. So you are putting this, you are putting this. These are the steps. Interpretation, as I have already told you, there are two types of organisms: acid fast organisms and non-acid fast organisms. Acid fast, which are red at the end, and non-acid fast, which are blue in the end. See the diagram. This is the diagram for acid fast. Can you see red colored rods, bright red color bacilli rods in a blue background? The background is necrotic, that's why to blue color. And these are acid fast, that's why retaining primary stain carbol fuchsin. They remain red only. They remain. So the same diagram. Can you see these red colored rods? The red colored rods in a blue necrotic background. So these are Mycobacterial tuberculosis. Principle I have already told you. So cell wall of Mycobacterium is impermeable to stain. Only carbol fuchsin will enter inside them. And after going inside, due to high concentration of lipid in the cell wall, it cannot come out. It will remain inside only. Cell wall become impermeable to carbol fuchsin and permeable for going inside, lekin impermeable for coming out. So it will not it will not be decolorized. That's why it is known as acid fast. Acid fast. Fastness. Fastness means resistance. Resistance is due to high concentration of lipid, a fatty acid in the cell wall known as mycolic acid. Very important. Now name the organisms. Name all the organisms which are acid fast. So there is a mnemonic for it. You already know Mycobacterium is one of them. But apart from Mycobacterium, there is a list. So the mnemonic is in front of you. I will tell you the mnemonic. The mnemonic is no longer separate booking for RIM SIM card. This is the mnemonic. How it is the mnemonic? Please mark it. Mark the mnemonic. I will tell you the mnemonic. So no ka N is for nocardia. Longer ka L is for legionella. Separate ka S is for smegma bacilli. Booking ka B is for bacterial spores. RIM. RIM ka R is rhodococcus. I is isospora. M तो सबको पता है. Everyone knows M is mycobacterium, both tuberculosis and Lipri. SIM का S is sperm head and card ka C is Cryptococcus. Now some of them are bacteria, some of them are parasites, and some of them are others, that is sperm or spores. So इनको अलग अलग निकाल सकते हैं. We can take it out separately. So you can see here how many bacteria are there, how many parasites are there, and how many others are there. Bacteria में Mycobacteria, Nocardia, Rhodococcus, Magma and Legionella. Parasites में Cryptococcus, Cyclospora, Isospora, and Taenia saginata also. Others में spores and sperm, human sperm, male sperm. So you can learn like this also. You can learn with the mnemonic also. So mnemonic is easier way to learn. Many MCQs are framed from it. Which of the following is acid fast organism? Which of the following is not acid fast organism? So that was about Ziehl-Neelsen staining. Now I am coming on modified Ziehl-Neelsen staining. What modifications are there? Modifications are there in case of percentage of sulfuric acid. As I have told you, normally we use 20%. Here in case of Mycobacterium leprae, we use 5%. Please learn percentage. For actinomycetes, we use 1%. For nocardia, we use 0.5%. On cryptospora, isospora and spores, we are using 0.25%. तो 5% किसके लिए, 1 किसके लिए, 0.5 किसके लिए, 0.25 किसके लिए, और others बाकी सबके लिए 20. So 20% is the standard. 5% for lepra, 1% for nocardia, 0.5%, sorry, 0.5 for Nocardia, 1 for actinomycetes, and 0.25 for Cryptospora, Isospora, and spores. You have to learn it. Many MCQs on this modification. So all these organisms, that is sporophytic mycobacteria, Lepra, actinomycetes, Nocardia, oocytes of the parasite, and spores, they are stained by modified Ziehl-Neelsen stain. So modification is by changing the percentage of concentration of decolorization. So as I have already told you, Mycobacterium lepra requires 5% H2SO4 for decolorization, and Mycobacterium tuberculosis require 20% H2SO4 for decolorization. So can I say Mycobacterium lepra is less acid fast as compared to Mycobacterium tuberculosis? Yes. So this is true. Very important MCQ. Mycobacteria lepra is less acid fast because it got decolorized only by 5%. It does not require 20%. So it is less acid fast. You got it? Now compare it with gram staining. Acid fast staining is done. What is the difference with gram staining? What were the 4 steps of gram staining? Here the primary stain was violet. यहाँ पे in acid fast the primary was red, carbol fustian, or secondary was violet, that is methylene blue. Here the opposite is occurring. Here primary is violet, gentian violet or methylene blue, and secondary stain carbol fustian, counter stain is red. And moderant यहाँ पे heat था, यहाँ पे iodine है. Decolorizer यहाँ पे acid था, यहाँ पे alcohol है, ethanol. So just understand it. So these are the 4 steps. Here primary stain is any violet stain. Gentian violet, crystal violet. यहाँ पे moderant is iodine, not the heat. यहाँ पे decolorizer is alcohol, and secondary stain is any red stain, usually carbol fustian or safranin. So primary stain, decolorizer, and secondary stain is changed. So you can see all bacteria are transparent in the beginning. You can see this is a transparent bacteria and this is also a transparent bacteria. When you put the primary stain, crystal violet, both become blue color. They took the color and they become blue color. By giving iodine, you are fixing it. It is acting as a mordant. It is fixing it. After that, you are putting decolorizer as ethanol here, not 20% sulfuric acid. So some bacteria resist decolorization. They retain the primary stain. Some undergo decolorization. They become transparent again. So after that you will put a counter stain, safranin or carbol fustian, some red stain. So the bacteria which retain the primary color, there is no space for secondary, so they will retain primary only and they will look blue in the end. And those who become transparent, now they will take the secondary stain and they will become red now. So the bacteria blue in the end are gram positive and the bacteria red in the end are gram negative. The difference is due to difference in their cell wall. You can see the 2 colors. Red color is gram negative and blue color are gram positive. You can see. So violet and red color, 2 colors are seen. So how many bacteria are gram positive and how many are gram negative? You have to tell me the list. So based on the shape it is either cocci or in the form of rods. 2 shapes are available. We know cocci is circular, bacilli are rods. So we divide. It is also gram positive, gram negative. It is also gram positive, gram negative. So we divide the bacteria into 4 categories. Gram positive cocci GPC, gram negative cocci GNC, gram positive bacilli GPB, and gram negative bacilli GNB. You have to learn example in all 4 categories. So the 4 categories are in front of you. You can see GPC, GNC, GPB, GNB. Have you got it? So what are the names of the organisms? So GPB, gram positive cocci, have only 2 bacteria, Staph and Strep. Important is only 2, Staph and Strep. Gram negative cocci, GNC, have only 1 main bacteria, Neisseria and Moraxilla also. So Neisseria, Moraxilla. Gram positive bacilli, there are 5 bacteria. The mnemonic is A, B, C, D, L. A for actinomycetes, B for bacillus, C for clostridium, D for diphtheria. A, B, C, D. A, B, C, D, L. L is Listeria monocytogenes. And gram negative bacilli are all. Except these, all of them are gram negative. That is how you have to learn. Again, the difference is due to difference in cell wall structure. Gram positive have peptidoglycan in their cell wall and gram negative have a thin layer of peptidoglycan along with outer membrane. So that is the difference. You can see the purple color. You can see the purple color here. Purple color here, it is gram negative. Peptidoglycan, it is thin, and in gram positive, you can see a thick peptidoglycan. So that is the thing. That is the thing. Last thing here is bipolar staining. A special type of staining is known as bipolar staining. Sometimes what happens now, can you see a cell here? All these are the cells. Where is the stain in these cells? Stain is accumulated at the 2 poles. So this is a cell. Stain is accumulated at the 2 poles. So it is looking like a safety pin. Have you seen a safety pin? So most of the students may already know what is safety pin. It is known as safety pin appearance. So stain is accumulating at the two poles. It is known as bipolar staining, number one, bipolar staining or safety pin arrangement, one and the same thing. Safety pin, looking like safety pin. So name the organisms which show the staining. So this is the name of the organism. Haemophilus ducrei, Vibrio parahemolyticus, not cholerae, Yersinia pestis, Campylobacter donovani, and Pseudomonas malai, Pseudomonas pseudomallei. So what is the mnemonic for it? There is a mnemonic. Safety pin appearance can produce very high yield. Can ka C is Campylobacter. Produce ka P is Pseudomonas. Malai, Pseudomallei dono. Very ka V is Vibrio parahemolyticus. High is Haemophilus ducrei or yield is Yersinia. So learn this mnemonic for bipolar staining. So many more MCQs based on it are there. So we will try few of them. So decolorization in Zn stain is done by which acid? 1% sulfuric acid, 5% sulfuric acid, 10% or 20%? Yes. I am asking about Zn stain. The standard decolorization. I am not asking about the modifications. Standard. Yes. Any guess? Yes. So the correct answer here is D. It is 20% sulfuric acid. Standard is 20%. Otherwise, we make the differences 5%, 1%, 0.5% and 0.25%. You know the examples. 5% is Mycobacterium leprae. 1% is Actinomycetes. 0.5% is Nocardia. And 0.25 is Cryptospora, Cyclospora and human bacterial spores. So these are the examples. So standard it is 20. Correct answer here is 20%. So bacteria with safety pin appearance, I have told you a mnemonic. So read the options and tell me what is the correct. H. influenzae, Vibrio parahemolyticus, Vibrio vulnificus or Salmonella paratyphi. Any guess? Yes, the correct answer here is B. So Vibrio parahemolyticus is there in the very high yield ka V hai ye. So I have just told you the mnemonic. Please see the mnemonic. What's the correct order in gram staining? See the four options. Very easy. So first primary stain, then mordant, then secondary stain. So primary stain is gentian volate, mordant is this is primary stain, this is mordant is iodine, and secondary stain is carbol fusion. So the correct answer here is A. Order is like this only. Gentian volate color of gram staining is due to what? In gram positive bacteria. Of course, it is due to cell wall में peptidoglycan content. Peptidoglycan content is more. That's why it is retaining the primary stain. So that is all about it. Thank you so much. If you like the concept, if you want more and more mnemonics, entire microbiology I will convert into mnemonics. If you want all the mnemonics together, if you want a short note on gram stain and Albert stain also apart from acid fast stain, if you want complete lectures on bacteriology, virology, mycology, parasitology, all of them are available for free. Where they are available for free? Where? On YouTube? No, they are not available on YouTube for free. My lectures. They are available on Unacademy Learners app. So go to the Play Store, install Unacademy Learners app from the Play Store, select a goal as NEET PG. After selecting the goal, you just search my name. My name is Dr. Priyanka Sachdev. See the spelling in the background. Dr. Priyanka Sachdev. You will find my profile link. Just click on my link. Follow my profile link. You will get all my recorded lectures at one place. So many micro lectures are available and you can hear the recording of all of them. So these are the free special classes. Free special classes. Why? What are the differences of what makes these free special classes different from YouTube? Why these are more important as compared to YouTube and better than YouTube? Because these are the interactive sessions. You can solve your doubts instantly. You can ask your question instantly and I can answer it. There is a poll option. So I will launch the questions in the form of the polls and you have to select the correct answer. There is raise a hand option. You will never miss the class. You will always get notifications 10 minutes before the class. And you will get PDF of the notes. So PDF of this acid fast stain, gram stain, everything is available. Why don't you now only go on the app and see, look for the PDFs and lectures, video recordings. It is available for free. And about the paid subscription is also available. There are two types of paid subscription, Plus and Iconic. In Plus, you will get access for live and recorded Unacademy lectures. From India's top educators, you will get live quiz, live test, printed notes, and more than 25,000 question bank assessment. And in Iconic, along with Unacademy lectures, you will also get access to Prep Ladder recorded lectures. I'm coming in a new batch in Target Next batch on the paid platform. The batch is started from 18th August. So all next aspirants should take the subscription and take the advantage of this batch. So we all educators together will teach you in this batch and prepare you for next 2023. We will teach you in an integrated manner. That is the specialty of this batch. Apart from it, few other batches available on the platform are Focus FMG batch, Target Next 2022 batch, and 2 batches for the NEET PG. So you can take the subscription for any batch. Apart from it, there is a NEET PG 1 month package available on the app. So only few days, last 20-25 days are remaining in NEET PG. So take this package. And the cost is also nominal. It is 4,500. And if you use my code SACHDEV10 over it, you will get additional 10% discount. The code is SACHDEV, SACHDEV10. Yes. And we will help you in preparation in the last few days. And we will help you to just ensure your seat in NEET PG. So we also have a clinical question bank in which more than 25,000 clinical questions are available. So you can practice, practice and practice. Two more last offers for you. If you take a subscription for 12 months, you will get 2 months extra. The offer is only for limited period. And if you take a 4-year subscription, the price of which in Plus is 60,000 and in Iconic it is 75,000. It is the most cheapest reasonable plan we are having, cost affordable and cost effective plan. The prices of for the 4 years. So all first year, second year students should go for this plan. Apart from it, we have many plans on the Plus and Iconic also. On the Plus starting from 2 months till 4 years. On Iconic starting from 12 months till 4 years. Longer the plan, cheaper it is. You can see the price. You can convert it into monthly installment of 1200 and 1400 also. Remember the code is SACHDEV10. You use any plan, Plus or Iconic. You have to use same code SACHDEV. SACHDEV, SACHDEV10, you will get additional 10% discount on all of it. The code is SACHDEV10. If you want it for free, you can try your luck, you can test your knowledge. I'm launching a scholarship test tomorrow. Tomorrow morning, I'm launching a scholarship test. It is on pathology. Systemic pathology questions will be there. I will give you 50 questions. The name of the test is Patho 50. The test is going to happen on 22nd August at 11 a.m. in the morning. Winners will get scholarships on the Plus subscription. So why don't you try your luck? Please, everyone, I request all the students to give the test. So tomorrow morning at 8:11, mark the date and set reminders for the time. If you have any doubt, take the snapshot, post it to us under respective subject. We will answer you. So there is ask of doubt feature on the platform. Our Telegram channel is Let's Crack NEET PG. Just join this Telegram group and you will get notifications of all classes. So follow my profile link. My name is Dr. Priyanka Sachdev. Go under the app, search my name, follow my profile link. So thank you. See you. Bye-bye. Study hard.